let's begin. The first thing we're going to do is on 4.1.1 on cell structure. Okay, so we're going to learn all about the cells and what do these cells have. We all know that the cells are the basic components of life, and we got that from Key Stage 3. So we're going to be developing that in GCSE. So the first thing we're going to learn about is actually eukaryotes and prokaryotes. And if you look at the first two learning objectives, essentially what they're saying is what are eukaryotes and what are prokaryotes and what are the differences between them, and that's mostly what sort of questions you'll get in exams. The last one talks about magnitudes of size, and I'll go on to what this really means and how does this apply back to this. So the 4.1.1 eukaryotes and prokaryotes, first questions that they could ask is what are the differences and similarities between eukaryotes and prokaryotes. So we look at the differences between eukaryotes and prokaryotes. Eukaryotes are things like us. We do have eukaryotic cells, and these eukaryotic cells make up plants and animals. Okay, and eukaryotes are usually multicellular. That means they have many cells, so we can say they're multicellular. Add. They're mostly multicellular. However, that being said, there are some that are unicellular. That's why I don't tend to make this big point here, because they both have they both have traits in both eukaryotes and prokaryotes of being some that are multicellular and some that are unicellular. However, the majority of eukaryotes are multicellular. And the big thing about eukaryotes and the distinguishing feature is this, the nucleus, right? We have an enclosed nucleus that contains all our genetic material. Okay, so if we just move on to looking at this picture here, right, we can see A is a prokaryote, whereas B is a eukaryote. And the distinguishing features can be seen already here. When you look at this nucleus, it's enclosed. That DNA isn't enclosed, it's free floating. So this doesn't have a nucleus. So no nucleus. The genetic material is what we call free floating. Okay. They also can have single DNA loops, or sometimes they will have these single DNA loops is also known as the plasmid DNA. But this DNA can also be free flow, like it will just look like a single, a double stranded DNA that's just there like that, okay? And it will just be free floating in that cell. Okay, so if we look at the relative sizes between eukaryotes and prokaryotes, we'll see that eukaryotes have a size of between 10 to 100 micrometers. Okay, we'll go into what micrometers specifically mean later on. The next size is below is prokaryotes are much smaller. The size is 0.1 to 5 micrometers. Now, if you look at the similarities between them, we can see that in both cases they have a cell membrane in. And some have cell walls in both cases. If you think about eukaryotes, look at plants, they do have a cell wall. Bacteria also have a cell wall, and prokaryotes are, a lot of them are like bacteria. But there are other different types of prokaryotes as well. And the other thing they both have in common is cytoplasm. And in the next video, we'll talk about more about what these are and what they exactly do. But just to quickly tell you, that cytoplasm is where all the sort of cell, like this is where the cytoplasm is. This is where all the reactions happen in the cell. Now, moving forward, we're going on to the other one. So they asked you to, in the learning objective, to know centi, milli, micro, and nano. So when they're talking about this, they are relating it back to something like the meter or the second. Kilograms is a bit of a weird one because this is all in relation to what we call the standard. These are standard units. Okay, so or SI units as we call it. So standard units or standard international units are the ones we use in physics and biology and chemistry to standardize all the units. And we use meters, kilograms, seconds, and there are others. But let's stick with meters and seconds as to be the easiest one. Kilograms we'll leave for now because you'll know the conversion rates of kilograms, I hope, but it doesn't work well with explaining this at this moment in time. But let's go through this. So centi means 10 to the minus 2. So for every meter, that centimeter, there's 100 of these centimeters that you need to make that meter. Okay, so we have 10 to the minus 2 and that's centimeters. And the millimeters is the same thing, but 10 times less. So 10 to the minus 3 is smaller. Micro is even smaller, it's 10 to the minus 6. And the nano is one of the smallest. There are even smaller ones, but the spec only went up to nano, so that's how far I'm going to go. Micro has the one with the weird unit, which looks like this. Nano, it goes back to just nm. This is just the symbol for micro meters. So to convert between these units, depending on which way you're going to times or divide. If you're going from a smaller unit to a bigger unit, you're going to divide. If you're going from a smaller unit to a bigger unit to a smaller unit, you're going to times. So here we're going to divide by 10 to the 3 to get from this to this. Here we're going to multiply 10 to the power of 3 to get to this to this. Now, you should have covered what powers mean in your maths lessons. If you haven't, I'm going to tell you right now. So 10 to the 3 just means that there are 3 zeros after it, because all this symbol is telling you, this power is saying 10 times 10 times 10, which is equal to 1,000. Right? There's 3 zeros, and that's all you need to really know about that. And if it's 10 to the minus 3, it's that, but you're doing it from decimal point of view. So you have from this 1, you go 1, 2, 3. So you go 10 to the minus 3. So because you've divided that 1 by 10 to the 3. So that's another way to write the same thing. It's the same thing. This to this is the same thing. Now it also asks you how to write in the standard form. So writing in the standard form, you're looking to write the number between 1 and 10. 10 is not included. So when it's less than, these are not included numbers. Actually, 1 is included. 1 is included. So if you wanted to add 0.001, how would you write that? 1, 2, 3, so 1 times 10 to the minus 3, because we take basically the decimal and we look at how many points does it take to get to this point. So it's 1, 2, 3. So it's 10 to the minus 3 because the decimal's here. So you have to go backwards. 1, 2, 3. But if you have to go forwards, let's say here. So if you have to go backwards, it's 10 to the minus 3. If you have to go forwards, let's put a decimal place in front of this number 1, 1, 2. So that's 1 times 10 to the 2. Same here, we put a decimal right in front of the first sort of digit. If it's a number bigger than the decimal point, then you go 1, 2, so it's 2.19 times 10 to the 2, so 1, 2. And so we take it from here instead. So we take it from the first non-zero number. So this is the first non-zero number, so we go back only 1. So it's 3.19 times 10 to the minus 1. Another example we can see, let's say we do 0.0419. Okay, then all we do is from this point, this is the first non-zero, we go 1, 2, so 4.19 times 10 to the minus 2. Okay, and let's go back to the spec. Looking at the spec, we can see that we have completed this section. We know that plants and animals have a cell membrane, cytoplasm, and genetic material in a nucleus, so that's the eukaryotes bit. Bacterial cells, which are prokaryote cells, are much smaller in comparison. We looked at the different sizes, and cells are usually in micrometers, right? And we talked about single DNA loops and plasmids, and there may be one or more rings of this DNA called plasmids. So these are the same thing. And I've also showed you the magnitude calculation as well. So that's all for today. In terms of now we're just going to go and so that you can stop here, know more from what you need to know from the spec. If you want to go further in the spec, I'm going to go ahead and explain that now. If you want to go further ahead and learn more about something, the one thing you can learn more about is standard forms and the different types of standard forms. You can also look at how kilograms work as well. So to further your development, what you can look at is into eukaryotes, are the different types of eukaryotes. Plants and animals aren't the only ones. You can look at fungi and all the other stuff. Prokaryotes, you can look at the other stuff as well. You can look at what we call the single-celled ones, single-celled eukaryotes, which are a lot more unique to these eukaryotes, and quite a lot more interesting, because this is like how evolution is sort of come about this eukaryotes of being single-celled over time, adapting and changing. And then you look at prokaryotes, and there are other things you can learn about prokaryotes in terms of look at the ribosome size. So if you know what ribosomes are, look at something called ribosome, look at the ribosome size, how are they different? So these are the extra little things you can look at, and other things you can look at are different organelles. Okay, organelles that are different. So I'm not going to go into that sort of detail. This is extra stuff. If you want to go into it, look at what the different organelles are, and just Google those things. But I don't want to do it in this video. It's just extras, so that's what it is.